today class, anatomy of flowering plants. In the last class, morphology, external features. But today is the internal structure. The internal structure of any organism is called as anatomy. Good. Root anatomy, stem anatomy, leaf anatomy is a differ from one organ to other due to presence of internal tissue. Now, anatomy, internal parts. Right. Now, what is tissue? What is tissue? A group of cells which are similar in origin and function is called as tissue. In plants, many types of tissue is there, but majorly two types. Meristematic tissue and permanent tissue based on division. It is ever dividing, it is never dividing. Right? The tissue which is present in the plant majorly classified into two groups based on a property of division. The tissue which is ever dividing is a meristematic. Never dividing, permanent tissue. But the permanent tissue formed from meristematic tissue. And that means the meristematic tissue is the source for permanent tissue. How it is, we can discuss later. Now come to now, what is the first type of tissue? Meristematic tissue. Now characters of meristematic tissue. Characters. Right. Be careful. Immature. The cells, before going to the maturation, is undergoes division. After the division, two daughter cells is there. These daughter cells, before going to the maturation, then is the division. That means, right, there is no sufficient time to maturity of meristematic cells. Hence, these are immature. Ever dividing without intercellular spaces. Without intercellular spaces. The cells are compactly arranged. The cell wall, most important. If the cell containing mature cell, the mature cell wall of mature cell containing three layers: primary wall, secondary wall, middle layer. These three layers together form cell wall, immature. But due to immature, the cell wall is there, but only primary cell wall. Right, now the vacuole is generally absent. Now, if it is present, small in size. Right. Plastic, which is helps in photosynthesis, chloroplast, is present, but in precursor stage, that is protoplast. Right. The meristematic cells not containing any reserved food materials. Right. The rate of metabolism is high. When compared with other tissue. Right. These meristematic cells are mother cells to all remaining tissues. That means in plants, all different types of tissues derived from meristematic. Hence, the meristematic cell is a mother cell to all other permanent tissues. Right. Now come to the classification. Whose classification? Meristematic classification. The classification of meristematic tissue based on origin, based on location, based on function. So, the meristematic tissue is having three classifications. One is origin, location, function. Now it is the based on location, where it is located. Right. Apical meristem, intercalary meristem, lateral meristem. Now, suppose you take one angiosperm branch. Now you know, no, it is the root tip, it is the stem tip, it is the branch tip. Good. All tips of plants is having meristematic tissue. This is called as apical meristem. Now, what is this intercalary. Right. Now this is the stem part. It is containing permanent tissue. Permanent tissue. Good. In between permanent tissue, if meristematic tissue is there, why it is called as apical? Apical. Who is apical? Meristematic tissue is apical. Why? It is present at the tips. Right. Like in the plant body, some meristem is present between the permanent tissue. That is called as intercalary. Intercalary. Sometimes it is present in the nodes, below the node and above the nodes. Now come to the lateral meristem. Now see, it is the stem. It is the stem. Suppose you have a permanent tissue, it is also permanent tissue. This permanent tissue is as separated by meristem. Now it is called as intercalary. Now another meristem is present in lateral regions. Is called as lateral meristem. This classification is based on location. Now apical meristem, intercalary meristem helps in vertical growth. Apical meristem, intercalary meristem helps in vertical growth or primary. But lateral meristem helps in horizontal growth or secondary growth. Right. Now the next one based on function. Protoder, ground meristem, procardium. Right. Now based on what is this protoder? If meristem forms epidermis, only epidermis. This is the suppose 300 meristematic cells is there. Among this 300, 100 stop the division, undergoes differentiation and form epidermis. So the epidermis is formed by a part of meristem. Such meristem is called as protoder. Another 100 they stop the division, undergoes differentiation and form vascular bundle, xylem. That is called as prokaryote. The remaining, they stop the division, undergoes differentiation and form mesenchyma, parenchyma, sclerenchyma. That is called as ground meristem. So this is the responsibility function. That means as epidermis, ground meristem, epidermis, ground meristem, vascular meristem. Now come to the based on origin. It is somewhat complicated. Just listen. Promeristem, primary meristem, secondary meristem. Right. Now, when you take the seed internally containing embryonal axis, that is embryo. This embryo containing meristem, embryonic meristem which is present in the seed is nothing promeristem. Promeristem, embryonic meristem which is present in the seed is called as promeristem. When you is called as primary meristem. Just catch the word. Primary meristem is formed by promeristem. Without promeristem there is no primary meristem. Promeristem means the meristematic tissue which is present in embryo or in the seed which is called as promeristem. Okay. When you keep in the soil, the baby plant. The promeristem remains. Some promeristem, not complete. Some promeristem, some embryonic meristem remains in the plant body throughout the life. Now it is the plant or embryo? Plant. But before plant it is seed. The seed with meristem or without meristem? Meristem. The meristem which is present in embryo is called as promeristem. When the seed is cultivated, some baby plant is there. During the formation of baby plant or primary plant body, the promeristem remains. Some not complete. Some promeristem remains in the plant body at apex. That is called as primary meristem. Right. Now what is the secondary meristem? The formation of meristem from permanent tissue by de-differentiation. Permanent tissues which are undergoes de-differentiation is having the ability to form the meristem. Such meristem is called a secondary meristem. Once again, secondary meristem. Secondary meristem. The meristem which is formed by the permanent tissue, that is specially parenchyma, chlorenchyma, but not sclerenchyma. Parenchyma, chlorenchyma. That means the parenchyma and chlorenchyma which is present in plant body, it is having ability, right? That ability is nothing de-differentiation for meristem according to its need. Such meristem is called a secondary meristem. Right. Now come to the right. Division. What is this? Primary meristem. Primary meristem and secondary meristem, which are present in plant body, when they are undergoes division and form permanent tissue. Once again, primary meristem, secondary meristem, primary meristem, secondary meristem. The meristem is always divided. By this division of primary secondary meristem, result is permanent tissue. That means collenchyma, parenchyma, sclerenchyma, xylem, phloem. Right. Now what is this? During growth of primary plant body, primary plant body, tender plant, young plant, right? During the growth of primary, apical meristem to whom? Plant. Which plant? Primary or secondary? Primary. Now, if the primary plant body, when it is going with growth, the apical meristem is responsible to form permanent tissue. Permanent tissue. Right. Now come to the permanent tissue. Permanent tissue. Right. The permanent tissue, it is never undergoes division. Permanent tissue. It never divided. Right. Based on its component. Right. Simple complex. If the permanent tissue is made with only uniform cells, uniformity is made with one type of cells, it is called as simple permanent. If the permanent tissue is made with different types of cells, it is called as complex. The name indicates simple. Simple, it is indicate complex, but both is belongs to the permanent because they never divide. Hence, this is the permanent. Why it is simple? Made with only one type. It is made with more than 2 types, or 3 types, or 4 types. Now come to the simple permanent tissue. Simple permanent tissue. Now, ground right. Right. A permanent tissue. Simple and complex. Simple permanent tissue is again based on cell structure, function, parenchyma, collenchyma, sclerenchyma. Now come to the characters of parenchyma. Right. If you take any plant, either monoplant or diplant, whatever it may be, it is problem. The major parts is containing parenchyma. That means the major tissue is major simple permanent tissue is parenchyma. Thus, it is the parenchyma is isodiameter. Length, width, height is same. Right. Shape, circular, oval, cylindrical, whatever it may be. Now, right. Large central vacuum. The meristematic tissue does not contain vacuum. Hence, the nucleus is present center. But here, parenchyma cells containing a large vacuum at the center, due to that the cytoplasm become peripheral. Now, as intercellular spaces may be present or absent. It is according to you. Centrocellular stress may be or absent. If it is absent, the cells are compact. Now function as parenchyma function, which is undergoes photosynthesis due to presence of chlorophyll. Such parenchyma which shows photosynthesis is called as chlorenchyma. Example, mesophyll tissue in the leaf. Right. You know, in dicots, the mesophyll is palisaded sponges. Right. This palisaded sponges is a parenchyma with chlorophyll. Hence, it is example of chlorenchyma. Now it is also storage of the food. Now it is also secretion function. The epidermis is present. Where the epidermis is there, that epidermis is externally covered by the cuticle. The cuticle is a secretion of parenchyma. Storage of food. Now this is the three important functions carried by parenchyma. Now, more important, now chlorenchyma. The cell wall is a much thick cellulose, hemicellulose pectin. At a time, I give the differences between parenchyma, chlorenchyma, sclerenchyma. All these three cells, cell wall is must be there. The cell wall of parenchyma, only cellulose. The cell wall of chlorenchyma, made with cellulose, hemicellulose, pectin. If the cell wall is made with pectin, blindly, it is chlorenchyma. Now what is another one? Sclerenchyma. Lignin. If the cell wall is made with lignin, the cell is nothing, dead. All cell walls in dead cells, cell wall of dead cell must be contain lignin. The presence of lignin in the cell wall is the character of sclerenchyma. The presence of pectin is the character of chlorenchyma. But cellulose is common to all. Now come to the chlorenchyma. Cell wall is much thick. Cellulose, hemicellulose is pectin. Without intercellular space. Now the chlorenchyma is present in hypodermis of dicot stem. It is most important thing. Hypodermis dicot stem. Dicot stem or dicot root. First of all, either it is dicot or monocot. If it is dicot stem or root, just think very deeply. Hypodermis, either monocot or dicot. If the dicot, stem or root, stem. The stem, dicot stem, hypodermis made with chlorenchyma. Now the chlorenchyma, it helps in mechanical support. For young plant and petiole, the petiole, it is having the resistance to wind. That resistance is nothing, some mechanical support due to chlorenchyma. Young plant, there is no secondary growth. The young plant is having mechanical support due to chlorenchyma. Now come to the sclerenchyma. Among these three, parenchyma, chlorenchyma is living, the sclerenchyma is dead. Hence, parenchyma, chlorenchyma is living, the sclerenchyma is dead. Hence, the cell wall without protoplast. The absence of protoplast appears as a lumen. Now, the most important in chlorenchyma, cell wall thickness. I forget this one. Cell wall thickening, without intercellular space. Cell wall thickening. Now, how it is? Just at corners. Now, it is one chlorenchyma, protoplast, a central nucleus. Now, it is another chlorenchyma cells. Now, it is the corner. Cell wall become more thick at corners. That is the important future of chlorenchyma. Cell wall much thickened at corners. This is the, be careful, keep in your mind. It is the important future of chlorenchyma. Now, sclerenchyma. The cells are long, narrow, dead. That means internally protoplast, cytoplasm, protoplast, all cell organelles are disappear. Right. The absence of protoplast appears as an empty cavity. The empty cavity is lumen. Right. Cell wall made with lignified. Right. Now come to this sclerenchyma is two types: fibers, sclerites. The fibers is the long, elongated, narrow, right, lumen, right, and it is also containing pits, plasmodesmata, right, sclerites, right. Now the sclerenchyma, hypodermis of monocostal, just here, collenchyma, sclerenchyma, collenchyma, okay, where it found in the plant, right? Example, hypodermis of dicotyledon. Now sclerenchyma, hypodermis of monocostal. It is hypodermis, but it is hypodermis of dicotyledon. Of course, the roots is not having hypodermis. Okay, I can agree. But hypodermis of dicotyledon is made with collenchyma. Right. Hypodermis of monocostal is made with sclerenchyma. Very important this example. Right. Now, sclerites. The sclerites is a spherical plate-like structure. Is one type of sclerenchyma. All right. Nuts. Fruit wall of nuts. Fruit wall, pericarp of whom? Nuts is example for the presence of sclerites. Pulp of guava, pear, sapota. These three fruits pulp contain sclerites. Right. Now, seed coat. You know seed coat, Tesca and tegmina is called as seed coat of who? Legume. Seed coat of legume containing sclerites. Leaves of tea plant. Right. This is the areas where the sclerites is present. It is very important. Now, right, the sclerenchyma gives the mechanical strength. Of course, collenchyma is also giving mechanical strength. It is also, but it is a living mechanical strength. It is a dead mechanical strength. So this is about the strength. Now come to the next. Complex permanent tissue. Simple permanent tissue is over. Simple permanent tissue, parenchyma, collenchyma, sclerenchyma. Now come to the complex permanent tissue. Based on function, this is again two types: water conducting tissue, food conducting tissue. The water conducting tissue is a xylem. The food conducting tissue is a phloem. Right. The xylem in flowering plants made with 4 different types of cells: tracheids, vessels, xylem fibers, xylem parenchyma. These 4 together form xylem. In parenchyma, not like this, only one type of cells, but here 4 types of cells. Hence, it is a complex. But among these 4, only xylem parenchyma is living. The remaining 3 are dead. Hence, xylem is the dead water conducting tissue, even though living is there. But majority, 75% of xylem made with dead cells. Only xylem parenchyma is a living. Hence, it is a dead water conducting tissue. In gymnosperms, vessels are absent. Among these 4, in angiosperms, water is conducted mainly tracheids and vessels. Among these 2, vessel is there. Right. These 2 are not involved in water conducting. They are helps and support to the water conduction. But among these 4, vessels and tracheids is a major water conducting cells. Right. In gymnosperm, vessels are absent. Hence, in gymnosperm, right. In the xylem of gymnosperm, water conduction by tracheids. Okay. Now come to the. Now, the vessels is a multicellular. Each cell is called as vessel membrane, which is arranged a longitudinal and interconnected. The ends of each membrane containing plates. This plates is containing perforation. Through this perforation, the water will be conducted. Now come to this xylem parenchyma. Xylem fibers. Xylem parenchyma is helps in storage of food and helps in is important. Radial conduction of water. Right. The water conduction is two types, radial and upward. Right. Now it is a radial conduction. Right. The primary xylem is again two types, protoxylem and metaxylem. Primary xylem, secondary xylem. During secondary growth, primary xylem, like vascular cambium from the secondary xylem. Right. What is this primary? Protoxylem and metaxylem. The protoxylem with small lumen towards peripheral. Whatever it is, peripheral or protoxylem. In all roots, xylem is exarch. Right. Why xylem in roots is exarch? The protoxylem is towards peripheral or central. Towards peripheral. Hence it is called as. Right. Now come to the phloem. It is a food conducting tissue. Right. It is also complex. Sieve tubes, companion cells, phloem parenchyma, phloem fibers. Now, among this, what is this? Phloem fibers is a dead. The remaining are life. Hence, it is living food conducting tissue. Named dead component of phloem fibers. Right. Now, what is this? Sieve tubes. Companion cells. The sieve tubes. Sieve tubes is a long cylindrical. It is another sieve tubes, long cylindrical, which are interconnected. Right. The ends of sieve tubes containing plates, sieve plates. The sieve plates containing pores, sieve pores. Now, what is this cell? Right. Cytoplasm. The most important. Right. Sieve tubes without nucleus. Hence, its function of sieve tube depends on companion cells which is associated with sieve tubes. That means where the sieve tube is there, neighboring cell is a companion cell. Right. Now sieve tube. Good. Where the sieve tube is there, its neighboring cell must be companion cell. Because the companions who are associated with what? Sieve tube is always associated with companion cell. Because the function of sieve tube is under control of companion cell. It is the main food conducting cell in the food. Right. Now come to the in gymnosperm, instead of sieve tubes, sieve cells, instead of companion cell, albumin cells are there. Right. The phloem of gymnosperm not containing companion cells, sieve tubes. Right. For this place, sieve tube place occupied by the sieve cells, companion cells is replaced by albumin cells. Right? The next. What is this sieve tubes? Sieve plate, right, without nucleus. Right. Now what is this? Without nucleus, it is a sieve. Good. The pressure gradient is important for the conduction. The function of sieve tube based on pressure gradient. The pressure gradient is regulated by companion cell. Pressure gradient. Right. Now, what is this phloem parenchyma? Is absent. What is this? Monocots. Right. What is this phloem? Phloem of monocot. is made with 3 components. Generally, the phloem is made with 4 components: seed tips, companion cell, parenchyma, and fibers. But in the case of monocot, the phloem of monocot is only 3. That is, phloem parenchyma is absent. Right now, come to the phloem fibers, which is generally called as bast fibers. As that is, the phloem is English word, Latin word is bast. B-A-S-T, bast. Right. Xylem is an English word, is the Latin word. In English is called as wood. Wood. The wood is an English word, common word to xylem. Now come to this next. Right. What is this? Phloem fibers, or which is also called as bast fibers, is absent in primary phloem. Primary xylem, secondary xylem. Primary phloem, secondary phloem. What is the primary phloem? Primary phloem, the phloem which is present by birth. But during secondary growth, vascular cavium, carcavium form the secondary phloem, secondary xylem. That means in young plant, which phloem? Primary phloem. In old dicot plant, right, secondary phloem is also there. That means the phloem is 2 types. Early phloem, primary. Late phloem, secondary. The primary phloem does not contain phloem fibers, is absent in primary. That means dicot. If you take the dicot plant, I given the phloem of dicot. When you see the microscope, right, phloem fibers is also there. That means the phloem is secondary. Because the primary phloem does not contain phloem fibers. Now come to the tissue systems. Tissue system. Right. Based on location. Right. In plants, either monocot or dicot, the tissue system is having 3 types: epidermal, ground, vascular. Epidermis, ground tissue, epidermal tissue, ground tissue, vascular tissue. Now, what is epidermal tissue? All the external surface of the plant, surface of fruit, surface of stem, surface of branch, surface of leaf, surface of flower, all the surface of the plant body is epidermis. This is epidermis, right? Some area, epidermis is having stomata. Right? Some area of epidermis is a trichome. Epidermis of young stem and leaf containing stomata. Epidermis of stem containing trichome, which helps in reducing of water loss. Cuticle. Stem of epidermis, leaf of epidermis, stem of epidermis, leaf of epidermis is covered by cuticle. This cuticle is also prevent or reduce the loss of water. Right now, root here. The epidermis of root containing a projection. This projection is called as a root here. It is helps in water absorption. Now this is the derivatives of epidermis. Now what it is? Stomatal apparatus. Which is helps in exchange of gases and also loss of water, transportation. This is the function of stomata. Exchange of gases. Atmospheric carbon dioxide is entering into the plant body through stomata. The oxygen which is released during photosynthesis, that is go to the outside to the atmosphere by stomata. But if the stomata is open, some water loss will be occurred. Now, stomata, God cells, substitutes. Now each stomata contains, stomatal apparatus contains as 2 kidney-shaped cells. This is God cells. Between the God cells, aperture is there. That is called a stoma. These 2 God cells is covered by substitutory cells. Now what are these kidney-shaped God cells? The most important cell is nothing. It is having 2 organs. Hence it is undergoes photosynthesis. This is all are modified epidermal cells on the surface of the leaf. Come to the Right. Ground tissue. Right. Right. In the stem, in the root, in the leaf. Other than vascular bundle, suppose what is this? In the stem. In the stem. Suppose. Right. Stem. If you take the TS of stem. Now, it is epidermis. It is hypodermis. Cortex. Endodermis. Now, pericycle. Cambium. Right? Xylem. Phloem. Now, this is internal structure. Right. Other than epidermis and vascular bundle, the remaining all areas is made with a separate tissue that is called ground tissue. That means the ground tissue as epidermal area, hypodermal area, cortical, endodermis, pericycle. This is an example for ground tissue. Right. Now vascular tissue. Vascular tissue. Xylem and phloem. In stem, in leaf, in the root. Example for gland vascular tissue. Now come to the anatomy of dicot root, anatomy of dicot stem, dicot root, monocot root, dicot stem, monocot stem, leaf. Right. Now, first of all, anatomy of dicot root. Right. For this anatomy, you must take TS. Now, it is a TSR. Dicot root. Right. Now, simple. Now, what is this upper surface area? Epidermis. Now, containing root hairs. Right? This is epidermis. So, it is which is tissue system? Epidermis. Now, below the epidermis, cortex. There is no hypodermis. Cortex is a multi-layers. Now come to what is the next one? Endodermis. Right. This endodermis is most important in dicot. Is having Casparian strips. Endodermal cells. Surface of the endodermal cell is having Casparian strips which is made with suberin. It is impervious, that is not allowed to the water. Right. Now the next one, pericycle. It is the pericycle. Now what is this next one? Right. Pericycle. What is the function of pericycle in dipora? Lateral roots and vascular cambium. This is what is this pericycle? It is endodermis. What is the importance of endodermis in the dicot root? The endodermal cells of dicot root is having Casparian strips which is made with suberin polymer. Now the next one, pericycle. Below the endodermal pericycle, they form the lateral roots and vascular. Below the pericycle, right. Now, xylem. Is it? Phloem. Xylem. Xylem. Like this. Now this is the phloem. The xylem and phloem is not present on the same radius, different radius. In the stem, these are present on the same radius. If the xylem and phloem is present on the same radius, like in stems, conjoined. But in roots, xylem phloem which is arranged alternately, hence such is called as radial arrangement. Between xylem and phloem, some parenchymal cells is there, that is called as conjugated tissue. Now come to what is this xylem strand? The xylem strand of dicot root has how many? 2 to 4. 1, 2, like this. 1, 2, 3, 4. Maximum number of xylem strands are 4. Minimum number is 2. This is the important character. Where the conjugated tissue we can see in the TS of dicot root between phloem and xylem. The arrangement is radial. Right. Now come to this. Now monocot. the same as it is, but the xylem strands is more than 6. More than 6. More than 6. The pith is well developed. Pith is, this area is made with parenchyma. The pith is present in dicot's root and also monocot, but monocot root containing more pith well developed. Now the differences between dicot root and monocot root. Dicot root, xylem strands, 2, minimum 2, maximum 4. But here minimum 6. Hence it is polyarp. It is tetra 4. Now come to the stem dicots. Now it is outermost layer, epidermis. It is trichome, not root hairs. Trichome. Below the epidermis, it is hypodermis. Whom? Dicot or monocot? Dicot stem. Right. Hypodermis means collechyma. Right. Cortex. Now below it is cortex. The next one is endodermis. The endodermis of dicot stem is also called as starch sheet because it stores the starch granules. Hence it is called as starch sheet. Below that it is pericycle. Right. Pericycle. Now like this. Now it is a pericycle. It is endodermis. The next one, vascular band. Vascular band is Now it is one vascular band. It is one vascular band. Like they are arranged in this vascular bundle of dicot stem, right, towards peripheral and external phloem, towards internal xylem and phloem is present on the same radius, hence it is called as conjoint and separated by cambium ring, hence it is a open vascular bundles. So the vascular bundles of dicot is described as ring, conjoint, open. Between two vascular bundles, parenchyma, that is medullary rays. Now this is the primary structure of dicot stem. Now come to the monocot stem. Right. In monocot stem, right. Now it is surface, epidermis. Now it is ears of monocot, right? Epidermis. Below epidermis, hypodermis. Right. It is made with sclerotype, but not chloride. Right. Now, right. Total cortex. There is no pericycle. There is no endodermis. Okay. Now what is this total cortex? Yes. Now this cortex is called as ground tissue. In this ground tissue, vascular bundles. This vascular bundles, right? Phloem, xylem. There is no cavity. Hence, it is closed and conjoined. Scattered. Peripheral vascular bundles, large in size than the center. It is the differences. This is all our vascular bundle. This is the vascular bundle without cambium, hence it is closed. This is the central, it is a peripheral, it is somewhat peripheral, it is a large, it is a small. Water containing cavities, this is a small cavity. Water containing cavities is present in the vascular bundles of monocot stem. This is the important feature. Now I am not saying the water containing tissue. It is a, right, what is this vascular bundle? In this vascular bundle, some space is there. Empty space. In this space, water is stored. Hence, it is called as water-containing cavities, where cortex, hypodermis, epidermis in the last order. That is the important feature of monocots. Right. Thank you. Now, anatomy of dicot root, anatomy of monocot root, anatomy of dicot stem, anatomy of monocot stem is completed. Now, as TS of dicot leaf, TS of monocot leaf, anatomy. TS of dicot leaf is also called as dorsiventral. Monocot leaf is called as isobilateral. Why it is dorsiventral? Why it is isobilateral? Okay, we can discuss now itself. The anatomy of dicot leaf, internal structure of dicot leaf is containing three regions: epidermis, mesophil, vascular bed. Right. Either dicot leaf or monocot leaf is having upper surface, inner surface. This upper surface, inner surface, lower surface, not inner surface, upper surface, lower surface is nothing epidermal region. That means this is epidermal, upper epidermis, lower epidermis. Upper epidermal is called as adaxal, abaxal, adaxal, adaxal epidermis, upper. Right? Right. Next one. So, what is this first area? Epidermal region. The surface is nothing epidermal. Between adaxal, abaxal, this is epidermal. Epidermal. See, it is adaxal. AD, it is between adaxal, abaxal epidermis, another tissue is there that is called as mesophil. Mesophil. The mesophil tissue in dicots again differentiated as right towards adaxal is a cylindrical, compact without intercellular space and which contain chloroplast. This type of mesophil tissue is called as palisade mesophil. Same mesophil tissue which is towards abaxal, spherical with intercellular space is called as sponge. That means in dicot, this total mesophil cell, in dicot, this total mesophil tissue is a differentiated two regions: upper region, palisade, lower region, sponge. It is the main tissue for photosynthesis in the leaf. In the leaf, name the parent, name the photosynthetic tissue in the leaf, mesophil. Right. The next one at the center. Not only center, where the veins is there, midway is nothing, xylem, that is containing vascular bundles. Right. Now, most important, just more stomata towards abaxal. Now it is a dicot. Now stomata is a more in lower surface. Right. Upper surface of dicot, stomata is almost absent. If it is present, less enough. So in monocot, we cannot differentiate which is adaxal, which is abaxal. That internal tissue is not differentiated. But in dicot, it is internal mesophil is differentiated. We can say this is a dorsal, this is a ventral. Hence, it is dorsal. Suppose you come to the monocot. Now, it is upper epidermis, it is lower epidermis, vascular bed, right? Now, total tissue is mesophil. Mesophil tissue is there, but it is a spherical loose intercellular space. Now, in this arrangement, it is somewhat difficult. Which is dorsal, which is ventral, which is adaxal. Hence, it is isobilateral. And stomata is also equal number. Here stomata is less or absent. Its stomata is more. Right? It is a palisade, it is a sponge. This causes differentiation, which is dorsal, which is ventral, hence it is called as dorsal. Okay. But TS monocot is isobilateral. That means bilateral, iso same. So adaxial, abaxial, which is appears same, we cannot differentiate because their internal anatomy is the same, not like dicot. Now stomata number is same, right? Mesophil tissue is also same, is not differentiated. But one of the most important in monocots, adaxial, this is adaxial, is containing specialized cells, is called as bulliform cells. Bulliform cells. Bulliform. These bulliform cells, when the bulliform cells absorb the water, the cells become turgid. Now, the leaf completely expands and exposed to the atmosphere. When these bulliform cells absorb the water, if water sufficient is not there in the soil or atmosphere, moisture is less, soil is also dry. So water content is less. Hence, right. What is this? Bulliform cells curl, folding inwards. This means the leaf is like this, curling. When it is curling, the surface area is decreases, rate of photosynthesis decreases. Hence, this is the important function of bulliform cells, which is present in monocots. Now, this is a dicot leaf dicot. Who is the dorsoventral dicot? Who is the isobilateral monocot? Now, come to the secondary growth. Secondary. Now, it is one of the important topics in the chapter. Secondary growth. It is only present in dicot. There is no secondary growth in monocot due to absence of cambium. Right. Here the cambium is there. The secondary growth in dicot stem, which is occurring only temperate region, geographically temperate region. The dicot plants which are present in temperate region, that plants show the secondary growth. The secondary growth results increasing width. The secondary growth is dependent on, this process depends on the activity of formation of vascular cambium, formation of cork cambium. First this will be formed. After that their activity. Formation and activity of vascular cambium. Formation of cork cambium and their activity. This is the two important events during secondary growth of dicot stem. Right, now it is a dicot stem. And the dicot stem anatomy. Now it is hypodermis, cortex, endodermis, pericycle, vascular bundle, phloem, xylem. Now it is called as medullary vesicle. Now it is a dicot stem. Now they want increasing their width but not vertical. Hence, it undergoes secondary growth. The secondary growth takes place by the two events, vascular cambium. Right. Now, what is this cambium? The cambium within the bundle or between the bundle? Within the bundle. Intravascular cambium. It is by birth. Hence, it is not secondary meristem. It is by birth. Okay. Next. What is this? It is also intravascular. Like this, the vascular bundles are arranged in ring-like. Now it is ring-like. Now, what is present between two vascular bundles? Medullary veins. Now the medullary veins which are present between vascular bundles is undergoes de-differentiation. Catch the point. Differentiation or de-differentiation? De-differentiation. Hence, now they have merged. Previously it is medullary waste. Which process is required? The conversion of medullary waste into meristem. De-differentiation. Now what is this? Cambium is also meristem. Now this is called as inter. Now this intervascular cambium, intravascular cambium. Now appears right here. Now this is called as vascular cambium. Vascular cambium. Now after the formation of vascular cambium, now what is the composition of vascular cambium? The vascular cambium is made with which tissue? Meristem. What is the character of meristem? Continuous evolution. Hence, this vascular cambium is undergoes division. The cells which are formed by this division, right? More cells fall inwards, right? Less cells fall outwards. Are you following? Now, what is this? Vascular cambium made with meristem. What is the character? Division. Hence, this meristems which are present in the cambium is ever divided. Due to ever divided, right? 70% of cells or 60% of cells, inside or outside? Inside. The cells which are formed, which are goes to the inside. Actually, what is this? Xylem. Xylem is there. Xylem. This is primary xylem. Now, these cells, new cells, which are going to the inwards to inside. Now, this xylem, primary xylem, already primary xylem, moves towards center. This place is occupied by the secondary xylem. Catch the point. Now, what is this vascular cambium? What is this xylem? It is previous. Now, it is undergoes division. Some cells fall inside, some cells is outside. The cells which are falling inside xylem. That means again it is a differentiated into xylem. Already xylem is there. That is primary xylem. Due to new synthesis of secondary xylem, the primary xylem moves to center. That means primary xylem towards center. The pith get the pressure. The pith gradually disappears. So, once upon pith and xylem. The xylem is primary, but it is a second time xylem is forming. It is primary xylem by birth. It is a secondary xylem, now it is forming. The secondary xylem push the primary xylem towards center. Already center, pith is there. Hence the pith crushed disappears. So, primary xylem. Now come to the outside. Now what is this? Phloem. Similar. It is outside. This is undergoes differentiation of phloem. Already phloem in the place of secondary phloem, which is present primary phloem. The primary phloem is goes to the outside like this. Right. The final pressure is reached the hypothalamus. Right. Meanwhile, right, pericycle, endodermis, cortex is degenerated slowly according to age, and finally, right, they get the pressure. Who? Hypodermis. When the hypodermis gets the pressure, now it is undergoes de-differentiation and form, right, carcocane. This is carcocane. Now, when it is carcocane, what is the cells? Meristem. Before that, it is hypodermis made with collenchyma. The collenchyma cells of hypodermis, when it received pressure due to activity of vascular cambium, then it is undergoes differentiation and form carcapine. Is it? A carcapine. Any cambium is made with meristematic. Hence again it will be divided. Now, this division cells is falling inside, this cells is falling outside. So, which are falling inside? Secondary cortex. which are falling outside calf. When the calf is formed, epidermis get the pressure and now the epidermis will be breakdown. Where the epidermis will be breakdown, that area is called as lenticels. Lenticels. That means, now what it is? Carpocalium. Carpocalium, which is called as phallosome. Now it is carp, phallum. Now it is secondary part, phallodum. Phallum, phallosome, phallodum. All together is called as periodum. See, right. Periodum. Last one. Right. Now come to the springwood. Right. Now, this is about the formation. But the activity of vascular calium and carcalium depends on physiological activities and external factors. Right. During spring season, this vascular calium is very active. Very active. Due to this, right. The secondary xylem is more and less lumen. Less lumen. That secondary xylem which is formed during spring season. Now, right, springwood and autumnwood based on factors, external factors. That the springwood is central with large lumen, right, light in color, hence it is light. Autumn, during winter, activity decreases, so the activity becomes slow, hence the lumen is right. The lumen is less and it is heavy. Lumen is less, that means internal cavity is less. Right. Hence it is a right late wood. Now, once again, springwood to autumnwood based on external factors. Right. Next one, hardwood and sapwood based on age. In every year, this is the dichotomous perennial. It may be 100 years or 200 years, maybe 40 years or 50 years, perennial. In 1 year, right, springwood, autumnwood, both are occur, which are appears like annual rings. One annual ring containing a circular of springwood, a circular of autumnwood. Based on these annual rings, we can determine the age of plant. Right. Now come to the, what is this? Right. According to age, hardwood, softwood. When the age is increases, irrespective of factors. Now central xylem, it is peripheral xylem, which is first formed according to your central is first formed. This central first formed xylem, right? Central inner xylem. What is nothing xylem? Right. Their cavities, xylem cavities, deposit organic matter. That organic matter, tannins, resins, oils, gums. Due to this deposition of this organic matter, the xylem fail the conductive tissue. Because the lumen is not empty. If the lumen is empty, that is when vessels, lumen of vessels is empty, the water upward conducting. But this vessels is filled with this organism. Hence, right, the central inner xylem, secondary xylem, is when they are filled with, when their cavity is filled with organic matters and not conducting water, but it gives the more mechanical strength. This is the character of central inner part of secondary xylem is called as hardwood. Now, peripheral xylem. When compared these two, it is a young, newly formed xylem. Secondary xylem both are connected. But their vessels is not filled with organic. Hence, they help in water contaction. Hence, this is called as hardwood and sap. Hardwood, sap. Both are secondary xylem. It is central, it is peripheral. Based on age, but not based on external factor. But once upon, it is containing annual rings. The annual rings is based on factors, external factor. Based on external factor, the secondary xylem is differentiated as springwood and right. This is about the secondary growth in dichord system.